Okay, where shall we begin? Well, this is team number 4, I believe. It's confirmed by Carla, by Carlo, by Fernando, and by me, Hector. And today we are presenting the research papers that we found and would like to expose as two possible things for our project. We already had a discussion with this, and we are somewhat centered on our project, but we're still defining it. And therefore, our research papers will have a higher degree of relatedness than if we just exposed what each one originally thought. Therefore, I think it is turn of, we will begin with Fernando or with Carlo. So can you please share your screen?
Yes, I'll begin. Okay, this is my presentation. It's called Cell-Based Antiviral Screening Against Coronaviruses, Developing Specific and Broad Spectrum Inhibitors. This article was published in 2013 by Loyola University in Chicago. It's a review article. So, it has 5 sections: introduction, druggable targets, cell-based screens for SARS-CoV, application and development of cell-based screens for MERS-CoV, and the current state of coronavirus drug development. First, the authors start to explain how do the MERS and SARS virus developed in bats, and then went through dromedary and palm civet, a cat, and then infected a human. And it states that there's not a single vaccine available for any of these two diseases, and the MERS outbreak was in 2012, and the SARS outbreak was between 2002 and 2004. This is a stat that is not presented in the article. The SARS has a 9.56% death rate. MERS has a 34.35% death rate, and COVID-19, the most recent coronavirus, is 5.76% death rate. So just to put in context. First, the article explains how does the coronavirus infects the whole cell and which of the targets are interesting to develop a drug for. So first you have the virus that needs to be activated, cut actually with the protein, with the protease. First, it has 2 ways. The first one is making a fusion with the membrane cell, with the membrane of the host cell. And the other one is just breaking apart and introducing itself to the host cell. Then it just starts translation. It produces its own proteins to make more viral cells, and then also has different replicase components. And curiously, one characteristic of coronavirus is making RTC, that is replication transcription complex, that is surrounded by a membrane vesicle called double membrane vesicle, DMV. So, but basically, this is all the targets, the most interesting targets that they found out about in 2014, and this is all you can try to target to end up the recent pandemic. Then, first we have the cell-based screens for SARS-CoV. We have the entry targets, that is the spike protein. Just try to mess with it. We also have the receptors, try to block the spike to rejoin to a receptor, or block the surface proteases to not be able to cut the virus to make it endocytosis. Or if it goes through fusion, block the caspins to not be able to cut the nuclear the membrane of the virus. Oh, wait. Then, why is it not working? Okay, here, okay. Then we have the polyprotein processing targets. It's also the proteins that need to be able to, the virus needs to be able to cut the other proteins. So it's like these are proteases that cut other proteins that the virus uses to make a ensemble of itself. And then we have the other targets, that is the replicase targets. These are different targets. These all are viral molecular molecular biology mechanism in order that allows to the virus to replicate. So these all are the targets, and the article states different methods that have been found in order to affect each one of these targets for SARS-CoV, and then goes more into depth to applications that have been developed in cell-based screening against MERS-CoV instead of SARS-CoV that were the ones in the last image. And then we have cell-based assays. We have two type of cell-based assays. We have infection virus BSL-3, that means working with the live virus, and we have surrogate assays that just use proteins or parts of the virus that are safe to use in a biosafety lab with level 2. These are just some of the ones that they did. You could try cytopathic effect, immunofluorescence, viral titer, that is calculating the amount of the viral particles infecting cells, QRT-PCR, reported viruses. So you also have spike serotype virus, in which you can try to block this spike, and protease cleavage bioassay. And that's basically the most, well, at the time, the ways that I have to try to study MERS-CoV. And then the current state of coronavirus drug development. At the moment, they had actually at this time they don't have an FDA fully approved vaccine for SARS or for CoV. For COVID-19, we're about to have it, but we don't have it still. And the article just tried to highlight the cell-based coronavirus screening and show novel methods of this screening. And the other purpose of the article is to assist in drug development for coronaviruses. And that's it. It's just a basic, really broad explanation of how coronaviruses infect the host cell, and how can we identify drug targets for it.
Well, fair enough. Any one of us has any questions to ask him?
I'm just asking if the both type of entries that you mentioned about the virus are simultaneous, or one is at MERS and one is at source. One about the external cleavage and one about the endocytosis.
It depends on the disponibility of trypsin. If you have trypsin, you go straight into the endocytosis, and then you have the membrane proteases that caught the coronavirus. If you don't have trypsin, it goes directly to target the receptors, and then goes through fusion. It just dependent of these.
So it depends more in the cell type.
Yep, it depends on the cell type. Any other question?
I believe not. So then we proceed with Carlos' presentation.
Okay, so in the article I want to present is the following one. It's called Biosurfactant, a COVID perspective. Is a review made by these authors, Matthew Smith, Stefano Phillips, and et al. So it starts with saying the characteristic of the virus, that it estimates that the R0 factor of this virus, aka the rate of it is infected, it's about 3.2. It means that one infected person can infect around 3 persons around itself. And the incubation period is variable, around 3 to 14 of incubation. It mainly infects the airways and the gastrointestinal tract, but it can infect other type of tissue around the body. And the severe complications of this illness are pneumonia and ARDS, that is the acute respiratory distress syndrome, that I will talk about it later. And it spreads using aerosols and droplets of coughing and sneezing. And the structure is composed by protein spikes, as Fernando mentioned in its article, that are the ones that give its name of corona, the lipidic membrane that surrounds the particle, the globular proteins inside the particle, and RNA for its genetic information. Then it talks about what are surfactants. Well, surfactants is a family of compounds. The bio prefix is about that they are manufactured by biological entities. And the surfactants are a group of chemicals that have the following structure. It has a polar head and a non-polar or hydrophobic tail. The polar head can be neutral, can be anionic, cationic, or a mixture of both. It's amphiphilic, as I told. It means that it has both philic structures, one that loves water, one that loves fats. It reduces surface tension, that's why it's named, between interface. These interfaces can be liquid-liquid or liquid-gas. And they're used also as emulsifiers in order to clean dirt, odors, and fats. They usually tend to have low cytotoxicity, which is beneficial for the use of humans, animals, and other type of eukaryotes. They tend to form mycelial structures that are like capsules, but this type of structures depends on a factor called CMC, that is the critical mycelial I don't remember the whole name, but it's a factor that means what is the concentration needed of the surfactants in order to make well-structured mycelia. And this, as I mentioned, it helps to encapsulate the compounds in the matrix. Also, it has antimicrobial properties, anti-adhesive, immunomodulation, and also anti-tumor. One of the two main families of surfactants are lipopeptides and glycolipids. In mammals, it's also been found that it can be used as molecular intercellular recognition for signal transduction, cell differentiation, immune response, and also in cancer, as I told, that has anti-tumor properties. And one disadvantage of this type of chemicals is that there is very few information about its effects in vivo, and that when it is used in surfaces of animals and plants, it could increase some opportunistic bacteria that feed or that live in these surfactants. But these surfactants are, as I mentioned, have antimicrobial properties. And how do they work? They damage or disrupt the cell membrane, and thus leading to a metabolite leakage, as you can see in the picture. This is an E. coli that's treated with SDSs. They affect the production of energy. The energy it's from having a charged membrane or by intaking something, some metabolite. It also disrupts cell transport and damage something called bacterial lipopolysaccharide system, or LPS, which it's a structure that helps bacteria to create biofilms. So if you attack this type of structures, then there's inhibition of the formation of these biofilms, thus cleaning more efficiently. And in the case of viral particles such as COVID-19, it is well known that the same property of amphiphilic structure disrupts the membrane of the virus, thus leaking all its internal content, the RNA and the proteins, making it inactive. And well, what are the known surfactants for health benefits? These are 4 types of families that are known to be effective in all the properties that I mentioned, and I will talk about it. The first one is the polypeptide family. As the name says, has a part of lipid and a part of protein. And these have antimicrobial properties. The ones that are commercial are the echinocandins, the micafungins, anidofungins, and daptomycin. These have today a patent and are manufactured by several pharmaceutical industries. Also, they have adjuvant properties which stimulate the immune system, and it's been tested that it can increase the effectiveness of cytotoxic T cells, thus increasing the immune response against some viruses. There's another family called surfactant, and this is the structure of the surfactant, and it has also immunomodulatory effects. It reduces the macrophage activity by blocking protein that is in its surface, that it's called the CD54. Also, it has anti-inflammatory properties by blocking some enzymes, such as the phospholipase A. And if you block this enzyme, you create no liberation of interleukins and nitrous oxide that are involved in the inflammatory process. There's also the family of sophorolipids. They are produced in yeast. If you inject them in mice, they inhibit some cytotoxins that may lead to inflammation and nitrous oxide in the case of sepsis. They can have also immunomodulatory properties, anti-inflammatory, and sepsis properties. And if you acetylize some of the structures, you can also increase the hydrophobicity of these compounds, making them real effective against herpes virus and HIV. So they can attack them like really quick and inhibit its entry to the host cells. And finally, there's cyclosporine. This is a well-known compound that is a biopeptide that is synthesized by this fungi. It inhibits influenza viruses by interacting with the synthesis of its proteins, and they module the immune system by a lot of metabolic pathways in the dendritic cells, macrophages, and neutrophils. Also, the article mentions its benefits in cleaning. Most of the cleaning products are around 15% and 40% of surfactants. Most of them are anionic, and they have the benefit that they cause no to low irritation and doesn't make chemical reactions such as bleach, ethanol, or hydrogen peroxide, which can be more efficient in order to treat some people and make no more harm than using this type of chemicals. And as I told you, there is the benefit against ARDS. ARDS, as you can see here, is an alveoli. And this is a healthy one, and this is the one that has ARDS. And when you have ARDS, it's a disruption. It's a stress of the structure. Thus, you have leakage of fluids, proteins floating, macrophages entering, some cells dying, and you have a lot of mucus. When you put surfactants, this review says that it can relieve the surfactant dysfunction, as you don't have the pneumocyte 2 cell that it produces surfactant. When you add surfactant, you relieve this type of tissue, making it like having something exogen that it can no longer produce because there's no more of these cells. If you have surfactant, also you can increase the O2 diffusion and thus decreasing the need of ventilation. And all these mucus, you can solubilize those substrates, making it easier for the lung tissue to clean all that stuff and facilitating the regeneration or the no longer damage of the structures. And finally, for pharmaceuticals, the use of biosurfactants it's related to generation of aerosols, which can be really useful in some drug delivery because they make mycelial structures that carry them safely. Also, these mycelia have the property to auto-solubilize, so you don't need to add some chemicals in order to absorb the wanted drug. And it's not only that thing, but complex of properties, because in its way to reach the target organ, it can inhibit some viruses and bacteria, as I mentioned. It coats tissue, which can relieve some inflammation and tissue damage. And it's also safe dosage and safe transfer for the drugs, which can be really promising in the case of COVID-19. And that's it.
Okay, so we have one question, time for just one question. Anyone ask? I want to ask, does it mention if producing these surfactants in different organisms cause a little bit of toxicity in cells or some sort of problem in separation, or directly if you apply it in some type of cell?
It's been said that there's no enough information, but generally they are low cytotoxic. And it more of the studies are most of the studies are in vitro, and it's needed to be wider range of tissue types and about in vivo studies in order to make these assumptions to be correct. Other question? No.
No, that's it.
I would just ask, does the paper mention any current research being added more into the COVID area, more recent research upon it?
Not really. It says that this field can have great opportunities, but doesn't mention if any of this type of surfactants are being proven today in the case of COVID-19.
Okay.
It's just that it's promising, but it doesn't mention if it's actually being doing.
Okay, Carla, you have any question?
No, everything is clear.
Okay, then we can proceed with your presentation.
Let me share screen.
Yeah, sure.
I can see where it's. Oh. I can.
If you click over there,
yeah.
this button, you can present.
Yeah. Sorry. Well, I'm going to talk about an in vitro tolerance test that it's made with erythrocytes in order to measure the potential of irritability or of surfactants. And these are the, yes, I can. So, well, a few introduction is that nowadays there are a lot of products that contain surfactants as raw materials in the production of cosmetics or cleaning industries. These surfactants can cause injuries and irritation mainly in the ocular membrane. So therefore, it is necessary to implement in vitro tolerance tests that at the same time it can be easy to perform and inexpensive to determine the concentration of surfactants that asserts the desired effect without having another effects or damage to other cells. So this in vitro assay is like an alternative method for stopping the experimental test with animals, and it evaluates the potential irritant effect of surfactants by quantifying the hemolysis and the naturalization of proteins. So they are using the oxyhemoglobin as an indicator, and they are spectrophotometrically detecting it, the exohemoglobin. The methodology consists in three main parts: the sample preparation, as well as the qualification of the surfactant, the hemolysis, and the denaturalization. So they made like different concentrations, different samples with different concentration of SDS, and they came to the conclusion that in the part of the qualification of the surfactant, it shows that the percentage was 19.2, which is within the specification and acceptance range. And in the graph 1, we can see that between 10 and 40 ppm of ECDS is the maximum point of oxyhemoglobin release, so that the concentration of ECD is, in which there is a maximum hemolytic activities on erythrocytes. So they graphed the absorbance versus the concentration of ECDS and came with the maximum hemolytic activity on erythrocytes. The results of this test established that the surfactant is between 24.1.4 with a 9.5 confidence of level. And regarding the denaturation results of the index ID, it came with the evaluation or the result of 1, and in expressing in percentage scale correspond to 100%. So it came to a conclusion that SDS is classified as an irritating position. And that's all. If you have any questions, please let me know.
Yes, I have one. So why did they use the wavelength of the spectrophotometer at 560 nanometers? Do you know?
In what part? In this?
Yeah. What were they measuring at that?
The absorbance. The absorbance.
But of what? Of what?
Of the samples with the different concentration of ECDS, and the samples with the erythrocytes.
Okay.
So what's giving its color? It's the hemoglobin, the oxyhemoglobin.
Yes. And also as a remark, I said that it gives a result of 1 corresponding to 100%. So it was an irritating position. These like scales are already predetermined or are already established. And I remember that you mentioned, Carlo, in your presentation that they are like limit between no irritating, irritating, or they are really irritating. So in this test, ISDS came in the conclusion that is irritating for the ocular membrane.
So what your article is saying is that this type of methodology gives the same results as other experimental methods.
Yes.
Okay. And one of my question is, this type of method can be, the results can be extrapolated to other type of tissues?
That's a good question. I really don't know in this case, but the European Union is making or developing new techniques, like I said, to stop experimental with animals.
Okay.
Well, if there is no more questions, I believe right now we have to make a little pause. In order to reestablish the session and continue recording it, after establishing it, I will be presenting the last presentation, and then that would be it for this group presentation for our project. So Fernando, if you, okay, now I will present my topic to you. Give me a second as I share my current screen. I think it's, give me just a second. Yes, here we go. Screen 2. Yes, let me, can you all see my presentation?
Yes.
Okay, well, the paper I wanted to present to you was generation of lung organoids from human pluripotent stem cells in vitro. Okay, give me 1 second. Well, the author's objective was to differentiate human pluripotent stem cells into ventral anterior foregut spheroids and further into distinct types of organoids, human lung organoids and body progenitor organoids. To put it in more general terms, was to present a possible model to study developmental biology and possible future uses of human lung organoids. They did the following protocol, starting from human pluripotent stem cells. They began a series of processes in order to set the cells through the use of different reagents as well as biochemical molecules in order for them to differentiate into the appropriate stage, at which they begin presenting it with step 15 to 20, right there where it says day 0 of culture. And the authors studied how to develop these two types of cells into the ones that are shown right here in the figure. The top one being human lung organoids, being air-like structures surrounding mesenchyme, as well as alveolar progenitors. This was done in a culture after 50 days. And the second part of the paper presents how this initial day 0 culture can be set into homologous lung body progenitors and how this can be set to remain in this state of quiescence, if it can be said, as long as it is desired for them to remain in this state. These human lung organoid trials were achieved as well in 2D as well as 3D with coordinating biological bioscaffolding in order to properly develop human-like organoids. Then they presented a table of comparison presenting the different processes that have been studied for the development of human organoids, as well as the time scale at which they managed to achieve these results. And we can see that some of them managed to produce alveolar-like structures, some of them with functional airway cells in vitro, as well as different models in vivo. Then in the results, as I have already mentioned to you, they managed to generate ventral anterior foregut spheroids at around the 10-day mark, and they managed to develop the different human lung organoids at day around 50 to 85. And they managed to let the bud-type progenitor organoids remain, well, for this to be produced in the 22 days mark. Then applications, the authors mentioned that they can use this type of models in order to study the developmental biology of these organoids in order to understand how these develop from embryonical state into proper development, into adult development, as well as these bud-type progenitor organoids, which have many possible uses, possible applications in regenerative medicine, in tissue engineering, as well as pharmaceutical safety and efficacy testings. Despite this, there are some limitations to it. One of the first limitations that can be found is that these human lung organoids take around 60 days to properly form and develop, which is a rather large time scale, and they are not highly expandable as other cell lines might be. Also, the lung organoids cannot fully develop into mature structures and cell types if they remain in media, even though they remain in 3D bioscaffolded media. They are not able to develop into fully mature structures. They require transplantation in order to develop to have the appropriate signals to fully develop. And the last problem they saw was that these cells exhibited a bias toward producing mucus. Well, the cells were mucus producing in the in vitro trials, and this is not quite understood yet. They do not make any specifications as though if this which is the chemical composition of this mucus. But despite this being useful or not for future studies, it is not well understood enough. And here's the bibliography. Do you have any questions?
My only question was, what's the difference between HLO and a bud type organoid?
HLO refers to human lung. The HLO refers to the human lung organoids. These are already a more differentiated type of cells than the progenitor buds. The difference between them is the differentiation state, one being more developed into pulmonary structures, while the other remains as a progenitors of this. Anyone else has another question?
Yes, what do they mean by transplantation? Like put it in a human?
Yeah, well, the trials for this for this full maturation of the cells was carried out in mice. And that's what the authors presented, that the full maturation of these types of cells was not fully achieved until they were implanted in an in vivo system. Despite the differentiation that occurs, forming alveolar-like structures as well as airway-like structures, which some of them do present the corresponding biochemical markers, these do not fully differentiate into the mature cells until in vivo transplantation.
So they did transplant it to mice and tested how they okay.
They did transplant it to mice, and that's what the others found out. What the others found. Anyone else has a question?
No.
Well, then if that's it, I think this is all for our first journal presentation. And for our second presentation, we will probably turn in a more specified project into how how different chemical products can be used with different cell lines to study how COVID can be prevented and treated, not just through a vaccine, but also through targeting the different systems that it attacks and how the disease can be attenuated as well as treated. If that's it, this is all from us.